In this video, we'll talk about the small and large intestine and begin to really wrap our minds around the importance of chemical digestion and what enzymes do to help break down our food. Immediately downstream from the stomach, really starting here at the pyloric sphincter, food in the form of that highly acidic and partially digested sort of soupy stuff called chyme leaves the pyloric sphincter and then flows into the duodenum or duodenum. It's the first 10 or 12 inches maybe of the small intestine. Then histologically, the lining of the intestines begin to change, and really a lot of digestion, chemical digestion and absorption takes place here in the jejunum, the middle half or something, middle third maybe. And then the last and largest portion is the ileum, and this contains a lot of lymphatic tissue. Here's the deal. Your gut, and by gut I really mean your intestines, your gut is a very busy, biologically active place, and it's not just your cells that live there. Obviously, you have a lot of cells in your own small intestine, including the cells that make up the intestine, the mucous membrane producing cells and the enterocytes, which are the absorptive cells. But the thing is, there are tons of white blood cells and tons of bacteria in your gut, and they're supposed to be there. I think we oftentimes have this idea that bacteria are bad and that bacteria make us sick. Yes, that's true. Some bacteria make us sick. But most bacteria don't bother us. Many bacteria help us to stay alive. And this is one of the most important lessons of this whole chapter. A healthy gut is like a rainforest. It is a very biologically diverse place. Hundreds of different species living in harmony help me to digest my food and help me to stay healthy. What happens if I take antibiotics to kill an infection? Well, oftentimes I damage the gut microbes too, and I might develop a secondary diarrhea or a secondary yeast infection. Some research shows that every time we go on antibiotics, we change the gut flora. We change the population of cells that live within the structure. Okay, so the whole point that I was trying to make is we have to have lots of lymph nodes there. Because remember, these are full of immunological fighters. So we want to provide a warm and happy place for our microbes to live, but we don't want to let them enter our body. They can stay in that warm tube, but if those fecal bacteria find their way into the bloodstream, holy cow, you're going to get sick and die really quickly. And so a healthy gut also contains a very robust immunological force, right? We got lots of white blood cells there, so they can keep the synergistic microbes in the lumen of the gut, and we can poop them out safely. But if there's a rupture, if the gut breaks open or ulcerates, the patient is going to become very ill very quickly. And it's just another reminder that, as strange as it sounds, this tube passes through my body, but it is not actually inside. The gut is the outside of the body. Okay, going back to the first portion of the small intestine, the duodenum or duodenum, we can see here there are tiny openings by which the pancreas empties its secretions. And so too, this green one, this is the biliary duct or common bile duct. So here's the arrangement. I'm flipping forward to slide 36. The liver makes bile. Bile is like soap. It helps to break down fats that you have eaten. In between meals, the liver releases tiny amounts of bile which begin to trickle down through these hepatic ducts and into the common hepatic duct and down through the common bile duct. And then right here, the doorway is closed. In between meals, We don't want to waste bile, so we store it up. This begins to fill, and it goes into the bile overflow tank, the gallbladder. Now, this perspective is a little misleading. The gallbladder does actually tip down and back on the underside of the liver, so that bile does actually flow into and then fills up the gallbladder. When I eat fats, especially during the intestinal phase of gastric function, the presence of acid and fats in the duodenum triggers the release of a hormone called the gallbladder mover, cholecystokinin, which triggers the gallbladder to squeeze out the soap, and this green sort of slimy secretion called bile begins to flow out. It neutralizes the chyme. It's a good buffer in its own right to help to bring the acid back up closer to neutral. But the most important function of the bile is emulsification. It shatters that big glob of fat into thousands of tiny microscopic fatty droplets. This is the pancreas. The yellow organ here releases enzymes and water and bicarbonate ion, the most important buffer in the digestive tract. So during the intestinal phase, we have to neutralize the acid and we have to emulsify the fats. And at this point, we need to introduce tons of enzymes to begin to chew up at the chemical level the fats, carbs, and proteins that we have eaten. The enzymes within the pancreas do all of those things. We have enzymes for carbs, we have enzymes for proteins, we have enzymes for fats. Now we have the bile working like soap to emulsify the fat so the enzymes can work on it. We have water coming out into the lumen from the pancreatic juice. Additionally, wastes from the liver are flushed out into the bile. Things like bilirubin, conjugated hemoglobin, and excess cholesterol. So if I'm eating a lot of dietary cholesterol, or if I have high cholesterol or fatty liver disease, my liver may be squeezing waste products out. Additionally, conjugated alcohol and toxins in our foods and metals to a small degree can also be excreted out through the bile. So here's a quick connection to your own healthy diet. A diet high in fiber, whole grains, beans, lots of fruits and veg. The fiber in our diet actually helps to trap those toxins right here. As that fiber passes through, it absorbs that excess cholesterol and those toxins, and we poop them out. If you don't eat much fiber, then those toxins just get reabsorbed right back in, and the liver has to try to do the whole process again. So that's the link between, one link between a healthy diet, rich in fruits and vegetables, and lower rates of cancers. It's also why Cheerios can claim to lower my cholesterol. The fiber in Cheerios, called beta-glucan, is particularly good at binding to biliary cholesterol. And so if you eat oats every day, oatmeal is probably good, maybe better, but Cheerios is fine. You're actually going to lower your cholesterol over time by binding to the waste in the bile and then pooping that out instead of recycling it. Okay, going back just a little bit. We see here the brush border. This is a very important concept here in the digestive chapter. The brush border is the surface of the lumen, which contains digestive enzymes, which help to break food down at the very last second. Let's see if I have a better picture here. Although this is the surface area of the large intestine, the small intestine looks very much like that. We see these little columnar epithelial cells. We could imagine that there are many little enzymes embedded into the surface of these cells. And as digestive fluids flow by, the enzymes begin to break those things down. Oh, I've gotten lost in my own PowerPoint. One quick second here. Okay. This is actually where the brush border is. In the small intestine, we have many layers of modifications to increase the surface area. You'll see that the interior of the small intestine is wavy. All of these little folds, the plica circularis, or circular folds, increase the surface area, maximizing the contact between our food and the layers of the cells that actually absorb the nutrients from the food. When we zoom in on the plica, or circular folds, we see that they are each covered in a fuzzy layer of tissues. These finger-like projections are called villi. And at this point, the soupy, foody mixture is flowing in between these fingers, slurping down in between, and as the intestines contract and squeeze, it circulates that liquid so that we have a tremendous contact and surface area between the partially digested food and the actual layer that will absorb it, which is this layer right here. This is the brush border. It is an epithelial layer of columnar cells. And if we look even deeper, we see that each of those individual columnar cells is lined with microscopic finger-like projections called microvilli. So here's the mind-blowing bit from this video. Although your small intestine might only be 20 to 25 feet long, it actually has the surface area of a tennis court, 70 square meters. How is that possible that I could pack so much surface area into such a small tube, you may ask? Well, there are 3 structural adaptations to increase the surface area: the circular folds, the villi, and the microvilli. And so if it were possible to actually flatten out all of these surfaces, what we would find is that our small intestine is just massive in terms of its surface area, and this is what allows us to absorb most of the nutrients from our food in just a few hours as it trickles down through the small intestine and then into the large intestine. The brush border enzymes, which actually chemically hydrolyze our food, are embedded within these microvilli structures. Too small to see, they are like tiny chemical scissors, such that when this protein comes down, the enzymes snip it apart into the individual amino acids, which can then safely be absorbed, ultimately entering the body here at the brush border, and then flowing into the bloodstream, where it can then be used to build my body. Likewise, carbohydrates, starches, and sugars are ultimately digested chemically into their individual simple sugars here at the brush border, where they can safely be absorbed into the bloodstream. So proteins and carbs are probably mostly digested already from the pancreatic enzymes, but any of that last little bit of snipping happens right here. Snip, snip, little fruit ninja, and then the tiny pieces go into the bloodstream. Fats, on the other hand, having been emulsified, have to pass through and into the lacteal. This green structure here is a modified lymphatic capillary, and these are where the little microscopic fatty droplets go, ultimately to the liver. Here we get a bit more detail on the different cells that we find in the small intestine. Downstream into the jejunum and ileum, we see most of our nutrient absorption taking place. The Peyer's patches are guarding for infection, and then ultimately the small intestine meets the large intestine here at the ileocecal junction. Going back to the brush border enzymes, these are a few of the specific enzymes we're going to need to know. Disaccaridases break down disaccharides like sucrose into the individual simple sugars. Monosaccharide means one sugar. For example, fructose or glucose. Peptidases break proteins down, hydrolyzing the peptide bonds, ultimately cleaving one amino acid from the others, so that we have to get down to this level, the individual amino acid, which can then safely be absorbed. Nucleases break down DNA and RNA. You want to review all of the anatomy of the liver for the lab practical, but pretty much everything you need to know about the liver we talked about here in the discussion of bile and the flushing of toxins. And then we see here some of the other functions. Carbohydrates are stored in the liver as glycogen. This is so important. We're going to talk a lot about this in the coming weeks. Glycogen is liver and muscle starch. So if I eat a high carbohydrate meal, my blood sugar will rise. Insulin will cause glucose to be put together into glycogen, converting the sugar into a starch. This is a win-win. Not only does that lower my blood sugar, because the sugar is leaving the bloodstream, but it also allows me to store that sugar for a few days in the liver, so that in between meals, or if I have to go for a quick little run, I can release the sugar into the bloodstream to keep myself alive. So that's a really important function of the liver. That function and dysfunction is definitely related to type 2 diabetes, something that we'll talk a lot about in the coming chapter. You can see here that the digestive blood, that nutrient-rich blood, comes from the small intestine into the liver through the hepatic portal vein, or porta hepatis, which we can see right here. Other important functions of the liver, which we'll talk much more about next week: nutrient interconversion. We can change just about anything into fat. I can change sugar into fat. I can change fat into fat. And in very extreme examples, I can turn amino acids into sugar. We also convert one amino acid into another. And all of that stuff happens in the liver. Obviously, we know that the liver is good for detoxification, and why people with substance abuse, particularly alcohol, tend to wear their liver out, because the liver is so busy trying to detoxify their system. Phagocytosis, lots of white blood cells here in the liver. And then the synthesis of many of our important plasma proteins and clotting factors. So you can see why you can live without your gallbladder, but you can't live without your liver, right? You can live without your gallbladder, because that's just a storage tank. But you can't live without your liver, because your liver makes bile, and it does all of those other life-essential things for you. In the event that you do have your gallbladder out, your liver can still produce a small amount of bile, so you'll still be able to absorb enough fats to keep yourself healthy. The problem is you're probably going to have to really cut down on the amount of fat that you eat, because if you run out of fat before you run out of soap, then you're going to end up with fat all the way down here in the large intestine. And in that case, you end up with a lot of smelly gas, potentially lipids in the stool, which would be a very uncomfortable position to be in. All right, backing up just a little bit. When I come back for our next video, we're going to talk a little bit more specifically about the gallbladder and the pancreatic secretions.